Good morning, everyone. Welcome to November 2025 Zoom Citrus Seminar. Thank you for joining us. First, I would like to thank Deborah for her help at our Hendry County Extension Office in LaBelle. Today's programs offers one CEU for pesticide license renewal and one CEU for certified crop advisors. If you need CEUs, email me your name, email address, and license number. The title of the seminar presentation today is Management of Common Citrus Diseases. Our guest speaker is Dr. Megan Dewdney, Associate Professor of Plant Pathology and Extension Specialist at the IFAS Citrus Research and Education Center in Lake Alfred. Dr. Dewdney will speak about disease management of greasy spot, citrus black spot, and phytophthora. She will give updates on the work she has been doing with greasy spot, re-emphasize the importance of CBS management, and discuss phytophthora management in the context of citrus greening or HLB. Dr. Dewdney, it's all yours. Please go ahead.
Thank you. So as Monty said, I'm going to be talking about several different citrus diseases. The first one I'm going to talk about is greasy spot. Just so that everybody has a refresher on the common symptoms. As you can see, we have these irregularly shaped black splotches on citrus leaves that do look a lot like motor oil as it gets into sort of an older stage, being sort of splashed on the leaf surface. Then we also have here on grapefruit the cloudy sort of nature of the lesions on grapefruit when they're minorly infected. Early season lesions with that yellow halos around the lesions or yellowing around the lesions on the leaf blade. Provided the leaves don't fall off, these level of infection, the leaves are liable to fall off, but provided those don't fall off, then the leaf will regreen back up, and then you will see it look a lot like this. And then we have some examples on grapefruit where greasy spot has gotten quite bad, and you can see that the rind has become completely discolored and in some cases a little bit depressed. And then these would not be suitable for sale on the fresh market, or at least not at a serious discount. And then when we get close up to the rind blotch symptoms on grapefruit, greasy spot is also called rind blotch. You can see that it's like little pinpoint spots of in between the oil glands, and that's actually the stomatal guard cells being injured. So the disease cycle is probably familiar to many people of greasy spot. Sort of starts, all the action starts just as we're coming into the winter season. So leaves that are starting to fall probably now with the cool temperatures, but all the way through to February is our major leaf drop. Those leaves will start to decay as we get wetter in the spring in the leaf litter. As those leaves are decaying, you get these patches of fungus where we have the structures that hold the spores called pseudothecia in the leaf litter forming. These hold the spores. These spores are then forcibly ejected during wetting events once they're mature. The spores of greasy spot are a little bit unusual. They don't actually directly infect the plant. What they do is they land on the plant surface, and then they grow usually on the underside of the leaf or on the fruit surface. This is called epiphytic growth. So epiphytic is a fancy word for on the surface of a plant. You can get a very minor number of secondary spores being produced called conidia. We don't really know if they have a major function in the life cycle, but it sure doesn't seem like they do. We then get infection through the stomates. That's where the guard cells are injured or die. And then the fungus then spreads out underneath in the mesophyll. and causing greater damage. And the epiphytic growth and the damage that you see on both leaves and fruit are closely related. And then you get the late winter coming around again, and generally you will see more leaf drop, and the cycle will continue. So it's one major cycle during the year. The symptoms will show up, early symptoms, particularly on leaves, will show up as early as September into December, but we don't often see symptoms on fruit at any intensity until sort of January time. But this is a little bit dependent on temperatures, and so if we get cool temperatures during the winter, then we don't see as much. But if it's warm during the winter, we will see it a bit earlier. As we start to see temperatures change over the winter months, we may be seeing a shift in disease symptoms, and we may have to revise our recommendations at some point. So inoculum production, as I mentioned, the primary inoculum is coming out of the leaf litter, but from those specialized structures. In this case, the primary inoculum of this fungus are the ascospores, which are a sexual spore. They are ejected during the wetting events. They form in the spores are partially in the partially decomposed leaf litter, and the wetting and the drying of the leaf litter is really important for the spore formation. So this is very old data at this point, looking at 1969, Jack Whiteside, my twice predecessor, collected data on spore ejections for this disease, and he found that the majority of the spores were being ejected in June and July, which very much lines up with the time of year that the leaves and fruit are susceptible to infection. Pete Timmer then found in the early 2000s that the cycle of spore ejections had actually advanced into April, May. And this was because we had improved the situation for the fungus in terms of maturation of its structures by changing our irrigation practices from overhead and pulled pipe to the microjets, which were much more efficient for irrigation, but they were also ideal for maturing these structures in the leaf litter. So that leaf litter maturation advanced the timing of the spores, and this was less favorable actually for the fungus because the majority of their ejection is actually happening before the leaves are particularly susceptible. However, because of its weird life cycle and that epiphytic growth, we do get enough spore ejection to allow for epiphytic growth to cause trouble even in these months, despite the fact that the majority of the spores have ejected earlier. So while it is good for us to be able to eject the spores earlier, unfortunately, the way the fungus works, it is able to overcome that by its epiphytic growth on the surface of the plants. So the big thing with greasy spot in the last few years is really being greasy green particularly on the East Coast. So as an update of this, I talked about it at Expo, but we were able to see that it says medium citrogreasium growth on the surface of the leaves. And we detected, not only on the leaves, but also on the fruit, we detected the fungus in a quantity from the leaf litter from our affected groves where we were doing our work. And we ended up concluding that greasy green is the same fungus that causes greasy spot. So while greasy green doesn't quite present the same way without the really plaque cast to the fruit, we are seeing enough damage to those guard cells that there's something about it that's causing the greening. So you can actually see that we do get damage amongst the guard cells, and it remains green in between the oil glands, and then finally with some molecular tools, we were able to say for sure that that's what's happening. So what is happening with greasy green? Why is this occurring? We think that maybe the time of year that infection is happening may be happening earlier because we are getting more erratic flushing with both the temperatures and HLB. And then we suspect that there may be some fungicide resistance contributing to insufficient managing. So where you might be thinking that you have good control, it's just not working the way you would have expected, and therefore you're getting this sort of lower level disease. What we do know through the work of Mark Reitenauer is that this problem cannot be resolved with post-harvest measures. We really need to manage this phenomenon in the growth. And that is the only way we're going to sort of keep this one in check. But I think if with some careful reconsideration of what our fungicides are and when they're happening, we should be able to get this back under to some sort of resolution. So, as I mentioned, we are concerned about fungicide resistance. We have some preliminary data showing that we have fungicide resistance to a common mode of action. We're working on the confirmation of that still, and we're looking at the extent of the problem as well. How widespread is it? And once I have more data on that, I will be sharing that with growers so that they can make improved changes into their fungicide programs. We are collecting a last season of epiphytic growth and greasy green symptoms. This is our last little bit to work on this project because we had some contradictory data between 2 years, and so we wanted to see which year was the correct year. Did it do one thing one year, and when we had data that did one thing one year and one thing a different year, we wanted to see which way it truly was going to be happening so we could make some more accurate predictions for the future. So that's what's happening on greasy green. I will hopefully have more to say about this in the not too distant future. We did do a rind blotch trial myself with the help of Mark Reitenauer's program in 2022. These were on white grapefruit in the Indian River region. Treatments were applied on June 7th, 28th, July 19th, and August 9th. We used a handgun at 200 psi. And then we collected the data in January of 2023, and we used a rating scale to determine how much damage was on the fruit. And so we had no lesions with a 0 all the way to a rating of 5, which is greater than 50% of the surface area. So we looked at 1, which is 1 to 4% surface area. These two ratings would have been considered to be marketable in our analysis. Anything above 5%, though, of the surface area was not considered to be a marketable piece of fruit. What did we look at? Well, we looked at double nickel with a surfactant cohere. This is a Bacillus amyloliquefaciens, and it's a biological, and we used 1 quart per acre. We also alternated that with Coside 3000, again at 1 quart per acre, with a 1.5% pounds of copper, and then double nickel with the Coside together, with the same rates as above, just at different timings. Looked at Coside 3000 in the new organic formulation, and we looked at 2 applications earlier in the season at 3 pounds, and then we used a reduced rate at 1 pound for 2 applications. We then also looked at Enable, which is a stalwart of greasy spot programs, alternating that with Coside 3000, and we used that at 8 fluid ounces per acre or 1 pound of copper. We looked at a newer product. They have registered this now for citrus. We were looking at it a greasy spot or a citrus with their proprietary surfactant K122. This is a different kind of product. Again, it's kind of a biological. It's based on the flagellar proteins of the BT peptide. We looked at it with 3 different rates, but only 1 application, 2 applications, and 3 applications of this project. We also looked at oxidate with cohere. This is a hydrogen peroxide and peracetic acid product. Looked at that at 1%, and then also at mixed instead of with a NIS, we looked at it with 435 oil at 0.5% and 3% of oil, and then we looked at oxidate with copper, and we looked at that at the 0.5%, and then looked at it along with 2 applications of 1.5 pounds of copper and then rotating to 2 applications of 0.5 pounds of copper. We also looked at 435 oil by itself, and then a newer fungicide from Syngenta called Moravis Top. This is an interesting product because it looks at difenoconazole, which is in Amistar Top, used to be known as Quadras Top, which is a good product, but they, instead of mixing it with a strobilurin, they're mixing it with an SDHI product, so pi-diflumethofen, and that was at 15 ounces. And then we compared everything to an untreated control. So what did we find? Well, all the treatments, fortunately, were better than the untreated controls. This is our average disease rating. Here's our untreated control. So everything is statistically better than that. 435 oil. 435 does a really nice job of keeping your leaves clean. It does not do a great job of keeping rind blotch off grapefruit. So that was what I would expect to see with this. I will say that in this trial, we did not see any disease on the leaves. We were supposed to rate leaves for greasy spot. We didn't see any, even on the untreated control, which was very interesting. Couple of our standards here, COSI 3000 does a fairly good job. Moravis Top looked pretty good. So did double nickel with Cohere alternated with copper, but our best 2 products were Enable alternated with copper and double nickel with the copper. The Aura Citrus, it certainly did have an effect, but it didn't have the effect we were hoping for. And Oxidate, again, also had an effect, but not as good an effect as we would like for a product. So our best performing treatments, as I said, were the Enable alternated with Coside and double nickel alternated with Coside. But all of these would reduce, all of the treatments that we tried would reduce your disease. Looking at the marketable fruit, all treatments had more marketable fruit than the control. So here is our remember that was categories 1 and 2. This is in percent marketable fruit. Here's our untreated control. You can see we had very few marketable fruit in there. The double nickel Cohere and Coside all mixed together gave us about 60% marketable fruit. Ideally, we would see more, but that's not bad. Enable alternated with Coside had slightly fewer, so more like 55%. These were not statistically different, though, from Moravis Top, double nickel with the Cohere alternated with Coside, and double nickel with Cohere by itself or Coside. And even the Oxidate had some effect with the Cohere. Where we didn't see great results were things like the oil. Obviously not very acceptable at 25%. So, but that is, as I said, what we would kind of expect to see with rind blotch. So looking at greasy spot management, our control products, really copper or all formulations, I think will work. We do have the strobilurin fungicides. I would suggest the caution for just a straight strobe, just in general. These fungicides are starting to sort of wear out their welcome. We're seeing more and more resistance problems here and there, and in all kinds of systems, and citrus will be no different. So those are Abound, Gem, and Headline. These are all straight products. Amistar's Top is strobilurin, is oxystrobin mixed with difenoconazole. I certainly would recommend that because it's a mixture. And then Pristine is mixed with is a strobilurin mixed with an SDHI. So with the strobilurin containing products, whether they're mixtures or not, I certainly wouldn't put them out more than once a year for greasy spot. We are concerned, as I said, about strobilurin resistance. That is under investigation currently. And our spray timing years really starting sort of probably late May to early June is for your first application for all of your different types of fruit, Valencia, oranges, grapefruit. That should keep it off the leaves as well. If you're looking at early oranges and grapefruit, then you're going to want to put out a second application in late July. And if you have had a lot of problems with either defoliation or rind blotch in the year before, you may want to look at an August spray, maybe with something that you could pair with either a brown rot spray or some other kind of application, disease that you might need management for. The fungicides that are, Enable is looking really good still in trials. It's a good substitute for copper if you're concerned with stippling, if you're in the fresh fruit business. Double nickel and Moravis top I thought looked really good in my trial, but I do want to say that I only have 1 year of data so far for either of these products, so it's hard to make a full recommendation, but at least in that 1 year they did look very good. Petroleum oil, we do see less consistent control on fruit than copper. You can mix it with all the other fungicides. Of course, you don't necessarily want to mix that with copper in hot weather because you are just lining yourself up for phytotoxicity. So do use caution with that. But I do know that a lot of the fungicides will work better with a little bit of petroleum oil, like a 2%. So what have we learned so far for greasy spot? So we know that the disorder greasy green that has plagued our grapefruit growers in the last 5 years has been shown to be Pseudomydium citrogricium, is the same causal agent of greasy spot and rind blotch. It just looks like it's a different manifestation of the disease, but we needed to show that. Degreening does not get rid of the green discoloration in fruit, and that control is going to need to happen in the growth. Our newer fungicides like double nickel and Moravis top look promising for rind blotch management. So as I said, but I can't make a full recommendation because we only have 1 year of data. Enable still is looking very good for management when rotated with copper. I certainly would be recommending that as part of a management program. So moving on to citrus black spot. I had thought that we would see a reduction in disease in the groves that we're working with in citrus black spot in the last year or two because we've had dry summers or drier summers, but we are already seeing disease starting to pop this year. So if you're out in the grove scouting, please keep that in mind. So we do see, so this is the typical symptom, the characteristic symptom of hard spot. Remembering that these lesions are less than ½ an inch. They usually present themselves as having a brown margin with necrosis on the inside or dead tissue. And then if it's a certain age of lesion, you will see with a hand lens, you will see tiny pinpoint dots, which are the structures for forming spores, so called pycnidia. And sometimes on ripe oranges, you will see a green halo. I did recently get a piece of fruit that had black or hard spot on it that was still green because it was a Valencia, and those lesions were actually had yellow halos on them. So that was kind of interesting because you don't see that very often. Crack spot is something we see not uncommonly in Florida. This is a symptom that's unique to the Americas. We think it is a marriage between rust mite damage and black spot damage, but you get this fine network of cracks, and it can cover very large proportions of a fruit surface. I think this one was over 50%, if I remember correctly. There are leaf lesions, but they're relatively uncommon. As you can see, these leaves have got some age on them. They're probably close to abscission, but you can see that these lesions look a lot like the hard spot lesions. They also have a yellow halo around them, and they can get pycnidia in them. The margin often looks sort of a cinnamon brown. This is one symptom that I have never seen yet in the field. I suspect it's out there. It's just I haven't been looking hard enough or something. But you can get lesions on the peduncles. As you can see, they're kind of hard to see because they don't have the margins on them. But these are out there probably forming spores. We get into the more virulent spot kind of things. These are the ones that are going to loosen your fruit and drop them on the ground. You can see this fruit was pretty heavily involved already. You get these large reddish brown lesions that are large patches, are very irregularly shaped, and then sometimes they go from something that looks like this to a hard spot, or they can coalesce and turn into this large patch. This is early virulent spot or freckle spot. You can see it's irregularly shaped. It's quite red. Lots of structures within these. and they can either turn into more hard spots or these can turn into those larger patches here or here. And then these are fruit that have got a lot of virulent spot here. You can see that the rind is just breaking down from the amount of infection on that. A little bit like you would see with anthracnose sometimes. So where is black spot these days? So the CHIRP program and FDACs are continuing to scout each year. But unfortunately, we keep having new blocks for them to being identified every year. It's still present in those 5 southwestern counties that were sort of early on. The Glades County was the most recent one that with quarantine zones in that southwestern sort of key core area. It's mostly in our commercial groves. However, the first residential find in Lee County was in 2019, and it certainly was present in that area for several years based on the symptom levels. And I think there's been at least 1 or 2 more residences that were found to have those black spot symptomatic trees as they came out of that, scattered more heavily in those areas. We also have 2 different locations in Polk now. So the hurricanes, we're not sure if they're spreading them, but it sure kind of makes you wonder. We have finds in Lee, Charlotte, and Glades County after Irma. Was that from Irma? It kind of followed the path of Irma's winds, but was that spread that had occurred before the storm and was just sitting in that undetected stage? We're not sure, and it's really hard to do the detective work to go back and figure that out. It's still too early to really know what spread has occurred because of Ian, but our newest finds do include Manatee, DeSoto, and Highlands County. So I think no matter where you are in the southwestern zone for sure, but even coming further north, it's best to be just vigilant for the disease and make sure that you are looking for the telltale signs of hard spot or virulent spot. So our current quarantine areas, got 9 counties with disease, including Collier, Hendry, Lee, Charlotte, Glades. We've got this one here in Glades, it's just bleeding slightly over into Highlands, Manatee, this location, original location in Polk, but more concerningly, this one here in Polk. So it is making moves, whether it is moving on equipment and vehicles, or whether it is moving naturally. I think in some of these further locations, I would suspect that maybe that's moving on human-made means, but it's going to be very hard to determine which is the case. So the disease cycle, talking about back to the disease itself. Again, this is a disease that a lot of things happen in the leaf litter. We get structures forming in the leaf litter as it decomposes. In most places, we see these ascospores forming in that leaf litter. They're ejecting, and they've got fruit or leaf on the fruit or leaf surfaces. Those spores germinate. They then penetrate straight into the tissue and go silent for months. You don't see a thing, which makes it hard to study. We get symptom development in the early winter or as the fruits start to mature. These then can form the secondary spore forming structures called pycnidia and conidia. Those 2 can infect, and then you get the whole cycle turning around. Well, that's the way it works in many places. However, here in Florida, we to date have still never found the ascospore, so we can just ignore that part of the cycle. However, we have also determined that a lot of our pycnidia are actually coming out of the leaf litter, which had not been shown outside of Florida until we showed that. So we get a lot. We still get them landing on the fruit or leaf surfaces. We don't know how it's getting from the leaf litter to the fruit surface. That's actually something we're working on now, trying to figure that out. We still get this latent phase, and the symptomology is all the same. And then as we get an application of the inoculum with the dead leaves on the orchard floor, so it's just sort of a little bit of a truncated leaf cycle, but it's something we need to be familiar with. So we only have one type of spore. I want to reemphasize that in Florida, it's the asexual form. They are splash dispersed conidia, so they're supposed to only move with water and not so much with wind. And nearly every other location with the disease has two forms of spores, the ascospores, the sexual spores that move on wind, and conidia. We know that these conidia are extremely abundant in leaf litter, and they're there all year. When they are in the canopy, they tend to move down more than splash up, which is logical when they move with water. But we suspect that rain splash is likely moving spores into the lower canopy from the leaf litter, and we don't know if it's using the leaves as it's asymptomatic to sort of springboard up into the higher canopies, because we certainly see black spot all the way to the top of the trees. So we only have the splash dispersed conidia. There are no ascospores. We see conidia coming from mature fruit, so obviously they're moving from fruit to fruit, when especially in something like Valencia, where you have the older fruit in there with the younger fruit, they can just drip spores down onto those younger fruit and get infected. We know dead twigs can have conidia, but when we were looking at dead twigs specifically, we didn't find as many conidia as I thought we would. We have found the majority of our inoculum in the leaf litter when we're actually looking for spore types. Most infections are occurring about 32 inches under an inoculum source based on literature and with that limited upward motion. So we have spores that are present in the leaf litter. They're splash dispersed, so generally they need rain to move. We don't know how much wind with our windstorms, with our tropical-ish weather, with the thunderstorms, how much that is assisting. We also have symptomatic fruit as an inoculum source while on the tree. As I said, we do have some inoculum in the twigs. Most older trees are at risk, but resets can be infected in 2 to 3 years in high inoculum growths. That was also not really sort of thought about in the literature. It said you didn't need to worry about black spot for about 10 years in a growth. However, when we were working down in the Immokalee area, we had a grove where we had mature trees, and then they were planting resets as they needed to pull some for blight. And those trees then started to show symptoms in 2 to 3 years. The fruit is most susceptible from formation all the way to September. Fortunately for us, April is often dry, but if you had a lot of rain, you would probably want to put out an application because the fruit are quite susceptible. All the fruit are vulnerable, but all citrus is vulnerable, but our late hanging sweet oranges are by far the most susceptible. When I was working in Immokalee area, we did have one grove where they had in close proximity a Hamelin and then Valencia, and the Hamelin almost never had symptoms on it, and I suspect that had a lot to do with the fact that the fruit were being harvested earlier and that it wasn't as the symptoms hadn't broken the latency period, but it may be that Hamelin was just less susceptible. The goal is to maintain. The goal with your fungicide sprays are to maintain coverage on fruit using minimum of once a month applications for best control. Start in May if it's dry in April, otherwise you should start in April. That's why I have that sort of bluish mark here, but otherwise May through September. If you need to rationalize your fungicide sprays, you're looking at your costs. If you need to prioritize fungicide sprays, I would prioritize them here rather than later in the season, because I think this is when they're most susceptible and they're most likely to have lesions that will cause the fruit to fall. So we did do a larger trial looking at the combination of skirting and fungicide timing. And what we wanted to know was, does skirting trees improve the outcome of fungicide programs? So here we have a very lovely grove where somebody's nicely skirted the bottoms of those trees, courtesy of Manji. And what we wanted to know also was, did applying fungicide treatments earlier or later than the current recommendations improve disease outcomes? So we looked at, we did skirting in our grove in June 4th in 2019, and then we applied fungicides with and without the skirting. We had two different sort of sets of dates in the 2 years that we did this trial. And then we had, but basically it went from early or mid-April, depending on the year, all the way to mid to end of October. And we looked at a standard fungicide program. So we looked at things like Abound and Coside 3000, so copper program in here, looking at another fungicide with an SDHI in here, as well as a strobe, Coside 3000, and then Abound again. When we had an early fungicide program, we added Enable at the beginning of the year in April, and then had the same program. For our late fungicide program, again, the main treatments were the same, but we added Enable at 8 fluid ounces again at the end of the year. And in our unrated control, we did not use a fungicide at all. So what did we find? So we looked at the disease in two different ways. We looked at the presence per tree. So did the tree itself have disease or no? Yes or no? So in 2019, 2020, we did not have a lot of disease in the grove, which sort of gave us sort of different results from the following year. So even in the control, only about 50% of our trees had disease. And in most cases, so we have blue is in no skirting and orange is skirting. So here we have our skirting treatment, and then we have our control, early, standard, and late fungicide treatments, and our disease incidence here. So there were no significant differences amongst the treatments, but you can see that particularly in the control, skirting made no difference. Skirting did seem to have a minor change for 2 treatments, but the result was opposite for the early, a little bit confusing. And then we did see that there was a slight difference in our fungicide outcomes, but the results were too variable to really make any conclusions. And then in 2020, 2021, you could see that almost every tree was showing symptoms, which was a little shocking to go from about 50% to almost all the trees. Here you can see a more consistent effect of the skirting treatment, but it's not large enough for it to be significantly better than the no skirting treatment. And none of our fungicides were stopping the presence or absence of disease on the individual trees per se, which isn't too surprising, because you'd only need one fruit with symptoms to have a disease that was present. So looking at disease severity, so how much disease was on the fruit, we looked at 50 fruit per tree. Again, we have our fungicide treatments: control, early, standard, and late, and orange and blue. Blue's no skirting, orange is skirting. And in 2019, when we had very little disease, so our rating here is we're only looking at a couple hard spots per fruit. You can see that skirting does seem to have a consistent effect for the incidence, but again, it's not large enough to be significantly different from the skirting treatment. When we look at the next year where we had a lot more disease, so here we have almost just a couple hard spots on each fruit. Here we go to where we have fruit, a large number of fruit that have about 20, 30 hard spots or a virulent spot on the fruit. So a lot more conducive for the disease. So skirting has an inconsistent effect all year long. Some treatments it helps, some treatments it doesn't. So you just can't make a conclusion from that. In this case, in each year, we do see a significant effect of the fungicide program. So we are reducing the amount of disease on the fruit with our fungicide programs, but it's a very heavy pressure year. And we do see a significant difference from the untreated controls in the early with the skirting, with both treatments, skirting treatments for the standard, and then with the skirting in the late season. So what is that? What did we learn from all of this? It was a tremendous amount of work. So we wanted to ask, we were trying to ask, did skirting trees improve the outcomes of fungicide programs? And I can say that skirting did not appear to improve our fungicide programs. We did see a minor effect. As I said, it wasn't significant, and only in low disease scenarios. So maybe if you only had very low disease amounts, maybe that could help. There are other good disease management reasons to skirt, getting in there with your herbicide, to better herbicide, making sure you're not knocking off fruit for brown rot, that kind of thing. So there may be other good reasons to skirt, but I don't think that black spot treatment should be your primary goal. And the other question we wanted to ask was, did applying fungicide treatments earlier or later than current recommendations improve disease outcomes? Those treatments in April or October did not appear to improve our disease management. With that higher disease pressure, though, obviously fungicide use was beneficial. It didn't really get rid of our problem, but it certainly reduced it. I would still say, though, if it was really wet in April, I would certainly probably put it on early. In the 2 years that we were working, April was very dry. We did have a late start to our trial, much later than I had hoped, at least 2 weeks. And that was because of the working with COVID restrictions at the university. But fortunately, our weather was quite dry. There was more rain in the summer of 2020 than 2019, and our relative humidity was elevated for longer in the year. And I think that might have contributed to the greater amount of disease. We did also collect the data in 2021, 3 weeks later than in 2020. This was based on our growers' harvesting cycle. We had, of course, to work with our grower cooperator, who was very generous in letting us do this big trial. And we did see very many more severe symptoms like virulent spot in 2021, which was a big surprise for us. So my last topic is Phytophthora. I guess I forgot to change this slide from just brown rot. But here we've got some brown rot. As a reminder, Phytophthora diseases are, of course, soil-borne, and they do travel into the canopy. These organisms are the causal agents of root rot, foot rot, and brown rot of fruit. We do know that yield reduction prior to HLB for all Phytophthora diseases was 3 to 6% per year, or approximately 20 million in crop losses. It's really hard to assess how much Phytophthora is causing crop loss now, but it's certainly one of the stressors on our groves. So all of our Phytophthora diseases are connected. Our root susceptibility is highest during a very wet to very dry cycle. So if we go from something that's really, really wet to all of a sudden into almost drought situations, that can really promote root susceptibility. That wetting and drying cycle, if we go from extreme to extreme, that can increase your root exudation. That attracts the zoospores. So as the roots get stressed, they're trying to grab nutrients, they're putting out exudates. These guys are in attune to that. They start to head towards the roots in their little swimmy ways. Phytophthora is more attracted to HLB affected roots than regular roots, which is concerning. And we don't know if that's because of a greater sugar content in HLB roots, because of the struggle with moving sugars throughout the tree with phloem plugging, but together we estimated or Graham et al. Estimated there was about a 27% root loss between the two organisms together, and Phytophthora alone was only causing about 10% root loss, which is plenty. It's enough to cause damage. So looking at our disease cycle, here we have the sporangia. They're releasing the zoospores. Those can either go to the roots and cause root dieback, or they can land on the trunks and cause damage to the trunks directly, or they can land on fruit that are on the ground or straight into the canopy. For brown rot, with rainfall at just the wrong time with the color break, especially if the temperatures are warmer, those decaying fruit can fall. They'll amplify the amount of inoculum there as long with infected roots. The Phytophthora are very much needing live root and fruit to propagate and keep their population going. So the mycelium in the fallen fruit and also from infected roots, that could turn into 3 different things. In some species, they're able to mate and form sexual spores called oospores, which then lead into the sporangia when the time is right. But it can also go into this resting stage where it I don't want to say overwintered, but it waits out the bad conditions. So in these resting spores, they're very well protected. They then also can germinate into sporangia or the mycelia can form sporangia directly, and then the sporangia form the zoospores, or they can infect directly themselves. So somewhat complicated disease cycle, but they're all interconnected. And you have to remember that if you have problems with root rot, then if you have a particularly susceptible variety like navel or Hamlin, then you may have problems with brown rot as well if it's wet at the wrong time of year. So brown rot particularly is problematic in early season varieties, navels and grapefruit, Hamlins. Navels and grapefruit, it's mostly in the packing house that we see problems with that, but Hamlin, you see it on the trees as well. This is because of a coincidence of rains with the color breaks. Most fruit loss is in those older, larger trees because you just have a lot of time for the inoculum to build up in the soil. If you're planting harvest, you can't harvest until all the infected fruit fall, and that's because of a phenomenon called nesting in boxes. So it's a disease that can spread from an infected fruit to other fruit around it, and it just spreads throughout the whole box, and then you just have to throw it away in a bin. So packing houses absolutely do not want that in their house. These can infect any type of fruit, but it's mostly on those early season fruit. We get that light brown leathery decay. So you can kind of see it here. It's not a great photo, unfortunately, but you do see some decay there. If it's humid, you'll see this mycelium underneath or on the surface, either underneath the canopy or in the packing house. They'll come in looking like that. If you've ever smelt brown rot, you probably won't remember it because they have that sharp, pungent odor that's just like, ugh, something is fermenting around here. What is that? And as I mentioned, it spreads post-harvest, so it's definitely something that the packing houses don't want to see. So we have two species of Phytophthora in Florida. That's Nicotiani, Phytophthora nicotiani, and Phytophthora palmivera. Nicotiani has got these lovely round sporangia with a little sort of point on the end. They are the common cause of foot rot and root rot worldwide, and they are a minor contributor to the brown rot problem. When we're talking brown rot, Phytophthora palmivera is really our main player. It's got these lemon-shaped sporangia, so they're quite visually different. It prefers poorly drained soils with high water tables, while that might be predominantly down in the flatwoods or on the coasts. You also have spots here on the ridge that can have problems. These tend to have very sporadic outbreaks. This organism is very sensitive to the wetting and drying. So if it goes through a dry cycle, then all of a sudden it disappears. It makes it really hard to study because it's not consistent, but it is a major brown rot concern, and that's because it can jump through the canopy. So brown rot control really should start with your cultural management. I don't think anyone's got a canopy that looks that good anymore, but if you did, let's hope you do soon. Then that's going to be really keeping the humidity in. You're going to see problems with brown rot. Trying to avoid premature fruit drop, that's going to be a real challenge in the HLB era. But anything that we can do to reduce fruit drop just to keep our fruit on the tree is going to also help with brown rot. Raising our tree skirts, as I mentioned, to increase air movement and promote the drying of foliage. We also have a low risk of movement, or you lower your risk of movement of the organism into the canopy through splash off those fruit on the soil surface. Your herbicide strip should just be inside the drip line, minimizing your soil splash, because it can also splash straight from the soil. That soil hits the fruit, and then you have inoculum on the fruit. And often recommended that a residual herbicide in early summer to sort of minimize the herbicide booms coming through and knocking off younger fruit from the equipment, which could then become inoculated. So you can see here we have this grove situation where the canopy is really grown in, and then you see that fruit from this particular grove has just gotten clobbered with brown rot. So we did a trial in 2023 looking at brown rot. This was in a 20-year-old Hamlin sweet orange grove on Swingle. It had a history of brown rot. This was actually down somewhere, I think, near Berea in Polk County. The treatments were applied with a handgun. We only put one application out on October 9th in 2023, and we collected fruit from the trees in day 1, 15, and 30 after application. We inoculated the fruit in the lab with Phytophthora palmivera to make sure we had some disease, and we incubated them for 15 days, rated them for disease, and we have in the treatments an uninoculated control to see the natural disease pressure in the grove. So we looked at Revis, which is a newer product from Syngenta, mandipropamid. It's got a fat group of 40, Verantis ultra, which is oxythiopiperine mixed with the mandipropamid, so a mixture of 49 and 40. I do want to mention that Revis is not registered for anything but brown rot control. It is registered for use in nurseries, so that makes it somewhat of a unique product. Verantis ultra is only registered for both fruit and root rot applications. And then there's Prophyte, a pretty standard treatment for brown rot, potassium phosphate. We looked at these at 8 fluid ounces for the two newer products, and then 4 pints for Prophyte, and then we compared that to an untreated control. So a relatively simple trial. And we looked for the presence or absence of brown rot, so incidence again. Here we have our treatments, and we have our brown rot incidence, and then our bars represent our days after the application, where we inoculated from day 1, day 15, and day 30. So here we have our untreated control. You can see there's a base rate of Phytophthora brown rot of about 22% in the grove. Changes a little bit over time. Not sure why, just the way things work sometimes. And then we have our inoculated control. You can see our inoculum worked a little better on the second 2 days. Again, sometimes that happens where you just don't get as many of the resting spores as you think you're going to get. A little bit inconsistent system, but between 60 to 100% disease incidence on these fruits. So they were affected or not. So looking at the actual treatments of interest, on our first day or day after the treatment, we kept the amount of disease with Verantis and Revis down to pretty much the same rate as we had in the untreated control. So the inoculations didn't take an effect. Looking at the second 2 days, you could see the inoculum had the effect of the fungicide is a little bit weaker. We were able to infect these in both Verantis and Revis. In the second day, it didn't work as well so having an effect here. I'm not sure why. We got a little bit more disease in that second day 15. And then Prophyte, we are seeing a reaction that we would expect. So remembering that Prophyte is a SAR inducer or a plant defense inducer, I shouldn't say SAR, plant defense inducer in plants, you would expect to see that it would improve with its efficacy over time. And again, we do see that. But if you have an emergency in your grove and you just want to get rid of Phytophthora right away, then I would probably look at one of the others. If you got a little time, then certainly this would be a way to go, because it does as well as the Revis and the Verantis. So we don't see any significant differences amongst the overall treatments. So when we looked at the percent of the fruit surface area affected, we see similar results. So we see here's our percent area of infected from 0 to 100, and then our treatments at the bottom. So we only had about 20%, between 15 and 20% of the fruit surface infected for the uninoculated untreated control. For our inoculated untreated control, you could see that disease we got more inoculation in that second and third date, but it looks a lot like what we saw in the is with the incidence. And here we have almost 90% of the fruit surface is affected. That means it's really hard to pick those fruit up because they're so soft and squishy, quite revolting. And then Verantis again and Revis were holding the disease back on the first day, a little bit less on the second days, but still having quite a different quite a significant drop compared to the untreated control. And then Prophyte, just it had the same pattern as before. We see less disease severity or less of the fruit affected with time. So while this wouldn't make a difference in terms of how you were, whether you had to wait for your fruit, it does mean that you're not getting as much inoculum production over time, which is not nothing. So for these treatments, the fruit surface area affected was less than the inoculated control, which is good. Obviously, our smallest lesions were found on our uninoculated control, and as expected, Prophyte got better over time. So with brown spot fungicidal management, often we're only got a single spray. Your best timing is to try and get it on that late June prior to disease symptoms, but I know that sometimes brown rot just, you're not expecting it, you haven't had a problem for quite a few years, and all of a sudden, oh my goodness, there it is, what do I do? But these ones in late July, we would recommend plant defense stimulating products, Aliet, Fostrol, Prophyte. They all have the same mode of action. You should get 60 to 90 days of control. If you're using copper before or after the symptoms are visible, you would use it in August. It's primarily protective like it is for everything, but it can kill inoculum, which is nice. So if you do have unexpected brown rot, then you might consider this in August. And then you get 45 to 60 days of control with copper. With the newer products, Rivas or Arandis, they're a good choice for late season if you've missed that earlier window. Here we would want to put out a Prophyte. Here we could consider copper, and then here we would maybe consider Rivas or Arandis. Usually, because if you're going to use it, then going to put out an application, then you really want to just stop it in its tracks as much as you can. We know that there's at least 30 days of protection. We didn't try and run it out as far as you could go. And we have added it to our recommendations for the production guide in 2024. So that was a recent addition. Unfortunately, for the root side of things, I don't have a lot of good data for these two products, or at least Arandis Ultra, I should say, not Rivas, because it's not registered for root application because of the challenges of working with HLB. So with young trees, my last topic here, I want to just talk a little bit about a trial that we did here at the CREC. We were looking at planting methods. Talked about this before. We had a bunch of different treatments. We had monthly insecticide treatments. We used metallicized mulch with or without insecticides, red dyed kale, and then individual protective covers. The only part of this I'm going to talk about today is our phytophthora results. And we took samples when we were looking at phytophthora, both near and distant from the irrigation emitters. And some of our treatments we did inoculate with phytophthora. So here, looking at our results, we planted in March 2020. First data point here was in August 2021. Our fibrous root density, which was the milligrams of roots per centimeter cubed of soil, and then we looked over time, looking at our different treatments here. In black, we had control, so this is how much roots we had. So both the control and the kaolin never produced very many roots until we see a little bit of uptick there. Our individual protective cover trees had a slow burn on root density, and then all of a sudden it popped up towards the end as those trees started to grow into their bags. And then we had the best root growth in our reflective mulch treatments. I'm not sure why the two of them were not consistent, but you get the idea that they were both growing pretty well underneath the reflective mulches for roots. So when we looked at our phytophthora propagules, so again, we're looking at colony forming units per centimeters cubed of soil and the same dates. There was no inoculation in this soil, but we had a fairly high population for coming out of having our old grove removed. Kaolin, unfortunately for the kaolin trees, they tended to have a large amount of inoculum to start with, but then it crashed down, sort of following the same pattern as the roots. As I mentioned before, if you don't have roots, then you tend not to have inoculum, but some of this might have also been just the inoculum matching the size of the root systems. But we see a similar trend in the control where we also had almost no roots. And the two of them fall down, the control and the kaolin, to almost nothing. A little bump up in the control towards the end of the project. Interestingly, we see a similar trend of that early decline in propagules with the IPC trees, but again, we don't have a lot of roots measured in that first year. However, we do see as the root systems increase, we see more propagules. We get a little fall down here at the beginning of the in November, but that may be seasonal because you see all the treatments except for the control do that. And then our two reflective mulch treatments tended to have higher propagules overall because they had roots. When we looked at where we inoculated, so we added even more phytophthora. We had 3 different treatments. We were interested in how much the reflective mulch was going to affect our root systems in general and whether that was going to change something for phytophthora. So here we have our fibrous root density. We then looked at just drip without the reflective mulch, and then the microjets as we've had traditionally. So in none of these trees had particularly great root systems, but by far the reflective mulch treatment had good root growth. And then we had sort of an improvement in root growth over time, but not much. And then you see the 3 treatments again, our inoculum population crashes because it's sort of coming to this, meeting the size of the root system. And then we see an improvement again in the population, and then we get a decline with the cool weather at the end of the season. But we do see more phytophthora propagules on this root system that is bigger. And these ones here, we see almost no propagules because there's almost no roots. When we looked at irrigation, so I put all of the treatments on this one slide. Here we have our phytophthora propagules at CFU per cubic centimeters, starting all the way from when it was inoculated in our first, I should say, our first sampling after inoculation. You can see here they're inoculated. They've got a higher number of propagules. And then we have our naturally infected ones. Everything falls down to almost nothing when it's far away from the dripper or the microjet. We just don't see any phytophthora at all. So you need water as well as roots for phytophthora. So young trees with healthy root systems are vulnerable to phytophthora still. Little has changed for vulnerable blocks with histories of phytophthora. If you're putting in new trees, particularly under the IPCs that are going to have nice root systems, you should probably be planning for phytophthora management if you have a place where you've had problems in the past. If you're not irrigating, you're not going to see a lot of phytophthora. So if you are sampling, please make sure you're sampling in the irrigated zone because we want to have an accurate representation to give you the most beneficial treatment recommendations. As I mentioned, if you have good root growth, you tend to see more phytophthora, especially if you're near the irrigation. Mulch helped the root density, so we saw more phytophthora there. And IPCs, trees under IPCs just tend to have better canopies and roots. So again, you're going to have more phytophthora. If our trees start to really flourish with OTC treatments, I again expect to see more phytophthora just simply because we will have more roots. Just want to quickly say some thank yous to all the people listed here on this slide who have helped in various aspects of these projects and different people on different parts. And I will, if we have time, I could take questions.
Again, if you are interested in CEUs, email me your name, email address, and license number. Thank you all for your participation and see you next month. Thank you.
Thank you.
Thank you.